Hello, everyone. My name is Iman Khwaja Ratib. We are pleased that you could all join us for this week's lecture in Volume 3 of our KnowNeuroanatomy didactic series that brings you lectures from experts in the field covering different topics each week. This series was created by trainees and early career neuropsychologists, including standing members of the KnowNeuropsychology Board, as well as members of the KnowNeuropsychology Committee. We would like to thank our 2023 sponsors for their generous financial support of this series. Additionally, we are grateful to our Society of Clinical Neuropsychology colleagues for their sponsorship and wanted to share information about their upcoming convention programming at APA in Washington, D.C. This will include 7 invited speakers, 9 symposia, 3 scientific poster sessions, a presidential address, and a member town hall. Registration for APA 2023 is now open. Before we start our lecture, one of the main goals of KnowNeuropsychology is to provide free, high-quality didactic content to our audience. Every KnowNeuropsychology and KnowNeuroanatomy lecture is available on our YouTube channel. Be sure to check it out, subscribe, and like our lectures to get first access to new content. New to KnowNeuropsychology is our collaboration with APSIN to bring you learning and discussion questions that are provided with specific lectures content. You can access these on our website and through APSIN. Here are the disclaimers for the series. This training is not meant to replace formal education in neuropsychology, and the views of the speakers are their own. Questions can be submitted via the Q&A box on the lower left of your screen, and a recording of today's lecture will be provided on our website and YouTube channel later this week. Now it is my pleasure to introduce Dr. Georgina Moreno for today's lecture titled Emotion and the Amygdala. Dr. Georgina Moreno is an assistant professor of psychology at the University of Houston, Clear Lake. Originally from Houston, Texas, Dr. Moreno graduated with a PhD in neuroscience from the University of Iowa and completed her postdoctoral studies at New York University. She also holds a graduate certification in multicultural education and culturally competent practice from the University of Iowa. Dr. Moreno's research is in the affective sciences with an emphasis on examining individual differences related to aging, gender, and race and ethnicity. More specifically, her research focuses on the effects of emotional states on cognition. For example, the effect of stress on decision-making, what variables may modify these effects, for example, sleep and social support, and the underlying neural and physiological mechanisms related to these processes. So now I'll turn it over to Dr. Moreno.
All right, hi everyone. So I thank you for that introduction. So yeah, my name is Georgina Moreno. I'm an assistant professor at the University of Houston, Clear Lake. And I'd like to thank the KnowNeuropsychology and the KnowNeuroanatomy committee members for inviting me today. I'm excited to share with you all a little bit of insights as our understanding of emotion and the amygdala. And so before I get started, so these slides will be recorded, right? So I wanted to put my contact information out there in case folks or if folks have any questions about what I'll be discussing today. My alma maters are up there. So my training, I completed my bachelor's of my BS in psychology at Texas A&M, where I did research in a behavioral neuroscience lab. I then went on to complete my PhD training in neuroscience at the University of Iowa. I was in the cognitive neuroscience program there, so the graduate program there. And I would say that my PhD training was unique in that I was in the Department of Neurology. And I was working or getting a lot of exposure to the Benton Neuropsych Clinic. My PhD advisor is a clinical neuropsychologist, Dr. Natalie Denberg. My program director is a clinical neuropsychologist, Dr. Dan Tranell. I got to attend our morning meetings 3 times a week where we would learn. It was a co-meeting with folks that were on the research side. So I was purely research, didn't really do any clinical training whatsoever as a student in the neuroscience program, but I got this really great exposure to both sides, so to both the research and the clinical side during my training at Iowa. And so I'm excited to share with you all a little bit of what I've learned during my training there. And then also through my training at NYU, I did my postdoc with Liz Phelps and did research there on emotion and decision-making, and also learned a lot about the amygdala and emotion processing. As far as currently what I'm up to, I'm an assistant professor at the University of Houston Clear Lake in the Department of Psychology. I'm also the director of our neuroscience program. So this is a concentration within our psychology master's program. It's more research track based than anything else. So most of our students go on to complete thesis projects, work in research labs. And I'm also the director of our Latinx and Latin American Studies program. So this is a minor at UHCL. So at UHCL, which is a teaching focused institute, I've really gotten the opportunity to continue my training, my work in neuropsych, in neuroscience and psychology, but also have gotten the opportunity to focus more on diversity, equity, and inclusion work, especially through programs like the Latinx and Latin American Studies program. So my email is there in case you have any questions or like to learn more. All right. So with that being said, so today the lecture that I have prepared today for you all is, what I'd like to do is go through a brief overview of how we study emotions in the contemporary sciences. And then I'd like to dive a little deeper and focus on one of the main brain regions that we like to discuss when we study emotions, which is the amygdala. And so just to get us started here, so emotions are studied in many disciplines, in psychology and neuroscience, but also in philosophy and sociology and anthropology. And over the last few decades, there's really been this emergence of the field, which we've titled, or which has come to be known as the affective sciences. And so in the affective sciences, this is the interdisciplinary study of emotion. And so in the affective sciences, we are asking, what is an emotion? How can we study emotions? And what are the functions of emotions? And so this is I know this is kind of really busy, but bear with me. So this is a figure from Dukes et al. In 2021 in an article titled "The Rise of Affectism." And so in this article, the argument was made that in addition to a lot of the core components of psychology in behaviorism and cognition, or cognitivism, as they refer to it in this article, there's been an emergence in the study of the affective sciences. And so this article, this was published in 2021, so fairly recently, puts forward the argument that in addition to studying behaviorism and cognition, we should also be considering the affective sciences independently as one of these core domains in psychology. And so under affectivism, or the affective sciences, we have topics such as emotion, but also empathy, so social emotions related to social processing. We have the study of feelings, of mood, of motivation. And so all of these different topics under this broader umbrella of affectivism. And so in this figure B here, this is an estimate of the NIMH budget share for funding in emotion research. And so what we see is that from the late 90s, early 2000s through the to 2019, 2018, we see that the funding in emotion research has almost doubled, right? So it's gone from close to 6%, hovering around 6% to upwards of 12%. Additionally, what this figure really points out is that we're studying emotion in many different contexts. So we're studying emotion in the context of behavioral research, in the context of attention, decision-making, perception, and memory, just to name a few kind of larger areas of research in which we're trying to understand the role of emotions or how emotion influences these different types of behavioral cognitive processing. Additionally, we're seeing an emergence of more formalized, specific ways in which we can disseminate research in the affective sciences. So we're seeing a growth in the number of affective science journals. So we have the Journal of Affective Sciences, the SCAN, Social, Cognitive, and Affective Neuroscience, the Journal of Affective Disorders. We have different textbooks that are being put forward and other books, and also a fairly new society. So we have the SANS, the Social and Affective Neuroscience Society, and the Society for Affective Science. And so with all of that being said, while we're seeing this exponential growth in the study of emotion, there's still a lot to be understood. And so there are many different theories of emotion, and there's surprisingly, or maybe not surprisingly, very little that affective sciences oftentimes agree on when we're trying to theorize how, what drives emotions, why do we experience emotions? One thing, or according to Scarantino from 2018, there are some general diagnostic features of emotions that most affective scientists can agree on. And so this is the concept that, or in general, most theories of emotion suggest that emotions are distinctive cognitive experiences or feelings, that emotions have distinctive motivational states. So we have emotions are feelings, emotions are motivations, and emotions have distinctive evaluations of the eliciting circumstances. So in general, when someone is in the affective sciences, when researchers are talking about emotions, we consider all of these different components that go into processing emotions. Additionally, what emotions, or what is involved when processing emotions, is that after we have some eliciting circumstance or something that occurred that leads to an emotional response or a change in your emotional state, we have first evaluation of that eliciting circumstance, and then also a response. And so the response can be physiological changes. So this can be changes that occur in the brain, changes that occur in our autonomic, in our nervous system, things that affect our heart rate, our skin conductance, right? So how sweaty our palms get, that things are physiological changes in terms of decrease in saliva flow, so feeling like you have a dry mouth, changes in the rate of respiration, so how we're breathing, even changes in our GI activity, right? And so additionally, in addition to these physiological changes, you also see expression changes and motor changes. So we see changes in our facial expression, in our body, in our voice, in where our eyes are focused to, how our posture is. Additionally, we have a response that is subjective, a subjective experience or a feeling that we perceive, right? So you feel hot, or you feel angry, you feel aggressive, you feel sad. And then finally, we also have a response related to our mental processes and behavioral disposition. So we have changes in our attention, we have changes in how we are engaging with others, with the world around us in that moment. And so all of these components are components of a response that we have to some eliciting circumstance, and that taken together can be considered an emotional process. In addition to these diagnostic features of emotions, we oftentimes study emotions can label the way in which we study emotions in a number of ways. So first, we might, a lot of the, especially earlier research in the affective sciences focused on emotion recognition, right? So emotion recognition being able to identify different emotions. So here we have Ekman's classic faces of different emotions being expressed, and so you're recognizing those faces. We can also study emotion in terms of emotion processing, right? So the way in which we process our emotions. We can study emotion expression, so the way in which emotions are expressed. And we can look at emotion regulation, so the way in which individuals regulate their emotions. So whether that be suppression techniques or cognitive reappraisal techniques, and so on. And additionally, with emotion, we often look at we can consider how we study emotion on different dimensions. Two of the most prominent ways in which we do this is by looking at valence and arousal. So valence being positive versus negative emotions, right? So on the spectrum from disgust and sad to happy and content, versus arousal, so high arousal versus low arousal, so the arousal or the intensity of that particular emotion. And so so even at its base, when we study emotion, it's very complex. And so it's not surprising that the network of regions that are linked to emotion and emotion-related processes is also very complex. So in the affective neuroscience world, there's a network of regions that are linked to emotion-related processes, including the amygdala, the ventromedial prefrontal cortex, the striatum, the anterior cingulate cortex, the insula, just to name kind of a few star regions that stand out. Historically, when studying emotion, we consider the role of the limbic system. I have limbic system here in quotation marks because the use of this term limbic system has largely fallen out of favor. So more and more we're moving away from using this term limbic system. Instead, we are focusing on the role of each of these individual brain regions and how they interact with one another. And that's another very important concept to consider, is that historically, when studying, especially in neuroscience, a lot of times we've historically tried to understand human behavior in terms of specific brain regions and what they might do, right? The amygdala is responsible for this or that. However, today we have come to understand that that's not how the brain works, that we very much need to consider the role of the amygdala in this more complex neural circuitry. And so this image here on emotional valence processing, so this is from an image from Underwood et al. 2021. And so in this diagram, what this study did was it was a meta-analysis, a systemic review of studies that were trying to trying to see what regions are most implicated when it comes to different emotional related processes. And so here, for example, this is emotional valence processing. We see that in red here, the most of the articles that were found in this review, the amygdala was mentioned, right, or was an important region in that particular article. Additionally, we see that there was a lot of studies that looked at the amygdala in connection to all of these different brain regions. So particularly the amygdala as it relates to the dorsolateral prefrontal cortex, the medial prefrontal cortex, and the hippocampus. Additionally, we also see a lot of connections within some of these regions that have nothing to do with the amygdala, right? So we see connections here between the dorsolateral prefrontal cortex and the anterior cingulate cortex, or the dorsolateral prefrontal cortex and the inferior frontal gyrus. And so highlighting again here that we have these network of brain regions that work together that help us to process emotional valence ratings, or on this next figure here, emotion regulation, for example. So this next figure, same article, this work by Underwood et al., where it's highlighted that the amygdala is another key player here, but then also the dorsolateral prefrontal cortex seems to be highly involved or represented in these studies that examine emotion regulation. And the idea is that there's this highly interconnectedness between all of these brain regions. And so with that being said, it's important to consider that today we understand that we need to consider these networks, or these neural networks of brain regions that work together to help in the involvement of emotion related processing. But with that being said, the amygdala is still a key player, and it has been for a very long time. And so these next few slides here, I just wanted to focus on the neuroanatomy of the amygdala. And so, so here on the left side, we can see the amygdala kind of sticking out from this medial section of the brain. So this is the right side of the brain, and you see the amygdala kind of sticking out here. And so the amygdala is in the medial temporal lobe, and it's right in front of the hippocampus. So if we were able to go directly into the cortex here from the amygdala and the kind of curve around, we would hit the hippocampus right back here, right? So if we were to go in, the hippocampus would be right back on the other side of these brain structures here. Additionally, I really like this diagram on the right, I think it does a great job at also helping us visualize where the amygdala is in the brain. So here we have, again, a medial cut or a medial view of the brain. And so here we have the right side of the brain, and then on the we have in blue, purple, pink, and green the left amygdala, hippocampus, and the basal ganglia here. And so this green little bulb here, this is our amygdala, right? And so we can see how it's this medial structure. So what's missing, so here they just popped off the cortex on the opposite side. And so it's a way for us to further kind of visualize where exactly the amygdala is. I think a lot of times folks struggle with, because it's this subcortical structure, right? Because it's deep in there, it's often very hard to conceptualize what that looks like. And then here's a few other images. So a lot of times we'll also see scans, right? A coronal view, a frontal view of the amygdala. And so here we're doing a cross section, or we're sectioning, doing a coronal section here, right through the amygdala. And when we look at the amygdala straight ahead, right in front of us, the amygdala looks like this. And then this changes, right? The further forward, or the further, the more interior or the more posterior that you go in these sections, this will change, right? It'll become smaller as you get towards the back and bigger as you get towards the middle, and then it will sort of start to feed into the front of the hippocampus here. And then additionally, with the amygdala, it's important to consider that it's actually a group of nuclei. So it's this collection of nuclei in the subcortical brain region. And so this is further trying to kind of illustrate that the amygdala can further be broken down into a number of different nuclei. And so this image here, bear with me, is another pretty busy diagram, but here just trying to get across the various nuclei that make up the amygdala. And so I really like this image here on the right. This is from Simic et al. 2021, where we see these different nuclei of the amygdala. And additionally, in this paper, they do a really great job of linking these different nuclei to specific processes. So these are different studies that have been done that have shown a connection between, for example, here, the basolateral amygdala and the striatum as it relates to reward processing and motor responses. And so we can see here the lateral amygdala, right, being related to a number of different, connected to a number of different brain regions, connected to a number of different brain regions and involved in a number of different processes, including interoceptive and exteroceptive processing here as it connects to the thalamus, for example. And so a really great image to study and to pull apart, especially I think it gives us a great general sense of just how the amygdala is, very well known for its role in emotion, but it's also involved in a number of different processes and higher cognitive functioning. All right, but with that being said, so the next few slides that I have here dive into our earlier understanding or the earlier studies of the amygdala, and some of the earliest links to the amygdala or some of the earliest links between amygdala and the emotion were through lesion studies in non-human primates. And so this figure here, for example, so this is a monkey who has bilateral ablations of the amygdala, so bilateral amygdala ablations. And it's hard to make out here because it's black and white, but they're openly, happily playing, or I shouldn't say happily, they're playing or interacting, also it's probably not playing, they're actively interacting with a snake. And so this study, these early studies in the 30s and later work by, so original work by Kluver-Bucy in 1937 and later work by Wes-Kranz in 1956, found that when these monkeys or when these primates didn't have amygdala, that their behavior changed. They no longer had a fear of snakes. But in addition to that, they also had a range of other very odd behaviors. So they would approach these objects that they feared, that they normally would fear, like snakes. They were orally exploring objects, so putting everything in their mouth, and then also exhibited very unusual sexual behaviors that they hadn't elicited prior to this brain damage. And then work by Wes-Kranz in 1956 found or kind of termed this particular deficit the Kluver-Bucy syndrome. And since then, so since this early work, the amygdala has really been one of the primary foci of researchers interested in the neural substrates or the neural systems of emotions. Work in the 90s, also lesion studies, but now in humans, also provided a lot of support for the role of the amygdala in emotion, and more specifically in fear processing. And so this case here, so the image that I have here is a brain scan here in the middle of patient SM. So this is a patient that presented with bilateral amygdala damage due to a disease called Urbach-Vitae disease, which led to abnormal calcification and led to this very pronounced damage that was pretty focal at the time to the amygdala. And so here we can see on the right image here, we see an intact, healthy comparison of what this might, where the amygdala should be. And here on the right, we see SM, where you should see some gray matter, some nuclei. We see these black circles, right? So very pronounced damage to the amygdala. And so here's another kind of very zoomed in image there. And so with this patient, they did a full workup on patient SM, and what researchers found is so with this damage, SM had a pronounced deficit when it came to fear processing, especially when it came to identifying when presented with Ekman faces, for example, a lot of difficulty with correctly identifying the faces that had fearful expressions on them. Additionally, patient SM she is unable to correctly identify fearful faces. So this research with her started, the first article on patient SM was published in 1995, and researchers continued to collect data on patient SM for 10 to 15 years. Her calcification, the Urbach-Vitae progressed, that damage did grow, and it did start to impede on other regions of the medial temporal lobes, but nonetheless, the findings with patient SM were particularly striking. So she again has this inability to correctly identify fearful faces. Additionally, she exhibited a blunted skin conductance response, so SCR. So SCR, this is right, how the conduction within her skin. So typically you'll put electrodes on the palm of the hand and measure signaling, electrical signaling there. And what was found was that where most folks would have some changes in SCR when viewing emotional stimuli, SM did not. Additionally, and this is from Adolphs et al. In 2005, SM also failed to use information from the eyes when identifying emotion. So in this eye tracking study, what researchers found, whereas on the left side here, we have control participants or comparison participants who would use information from the eyes to help inform the different emotions that they were identifying. SM very much averted, for the most part, the use of the eyes in order to help inform her of what emotions were being identified. So had very few fixations with the eyes, and this is significantly different from these comparison participants. Additionally, in 2011, work by Feinstein and colleagues conducted a study with SM where they took patient SM on a number of different experimental trips where they tried to induce fear in patient SM. So first, they had patient SM, they tried to see how she would react to handling a snake. And so this figure here, figure A, is the actual patient. This is patient SM here handling a snake, and indicating that she very little fear, if any, when doing so. This second image here is an image of a tarantula. So they presented SM with this tarantula, and right away SM tried to touch the tarantula, tried to pick it up, exhibiting no fear. And then probably one of the most interesting components of this study was they even took SM on a haunted house tour. So they took SM to the Waverly Hills Sanatorium haunted house. And they, again, through this experiment, found that SM was unable to elicit or describe feeling fear in any way. So again, suggesting that the amygdala is very important when it comes to processing emotions, and in this case, especially in processing fear. There's also been fMRI studies that have further confirmed a role of the amygdala in fear processing, more specifically in fear conditioning in humans. And so this is a large body of work in humans. And this is extension of work that started in animal models, but that extended into humans, where researchers use fMRI, so functional magnetic resonance imaging, to study fear processing and to investigate the neural correlates of fear processing, or in this case, fear conditioning. And so in these studies, participants are presented with blue squares, for example, and are through conditioning procedures are come to learn that when presented with a blue square versus a red square, they might receive a mild shock or stimulation in response to the blue square. And so they become conditioned to expect or they learn that the blue square might result in some in a shock, right? And so they develop this fear conditioned response to that stimuli later on, even when they're no longer being shocked. When you just show them a picture of a blue square, this still will exhibit a response to that stimuli. And so this is work here from Hannon et al. And work from Kevin Labar et al. And the Phelps lab et al. And so what researchers found here, that activation of the amygdala, so here we see so activation in the amygdala is able to predict later memory. So suggesting that the amygdala plays a role during encoding, or in the encoding of a subsequent memory. Additionally, amygdala, so here on the right side, we see amygdala activation to an episodic representation of fear, especially in the left amygdala. We see activation in the amygdala as it relates to these different fear conditioning paradigms. And so again, further suggesting that the amygdala plays some type of role, whether it be a modulatory role or something of similar when it comes to encoding memories, especially emotional memories. Additionally, the amygdala has been found to modulate many other facets of cognition, including memory, as we just discussed, also perception and attention. So there's research especially looking at different networks, so the salience network, and a host of other functional connectivity networks that often we see the amygdala implicated in that work. There's also been a lot of research looking at social stimuli, looking at processes like empathy, looking at when looking at social threat, looking at race. We see activation, or the amygdala seems to be particularly important or involved in that process. Additionally, when it comes to reward processing and the decision-making research, we have often seen, or there's a growing body of literature suggesting that the amygdala plays a large role in that, in modulating that aspect of cognition. And then additionally, there's been quite a bit of research that implicates the amygdala when it comes to major psychiatric disorders. And so this, excuse me. So this image here, this is from McTeague et al. From 2020, where they demonstrated there are so increased activity, and hypo, or so decreased activity during, or that demonstrates abnormal activation in the amygdala, as well as a number of other regions, including the fusiform gyrus, the ventromedial prefrontal cortex, the ventral medial, the thalamus, and a lot of our ACC, the anterior cingulate cortex. But here we see, strikingly, we see this very robust activation, or aberrant activation, right? So not typical, not what you would typically see in the amygdala. So both in the left and right amygdala, as highlighted in this frontal view, and then also here from this medial view, we see a left amygdala, and also extending into the hippocampus back here a little bit, activation. And so this research also suggesting that we see these patterns, or we're identifying these patterns of neurocircuit disruption across a number of major psychiatric disorders in regions and networks that are key to adaptive emotional reactivity and regulation. And so a lot of this has also been further linked to various types of networks, so salience networks, reward networks, non-reward networks, as they relate to, and how these different brain regions are connected to one another. And so, all right, and so with that being said, so with today's lecture, what I really wanted to do was to give you a brief overview of our understanding of the affective sciences, and what I hope that you take away, and the role of the amygdala, and also other brain regions. But what I want you to take away from today's talk is just consideration that together with a network of other brain regions, the amygdala plays a role, a very important role in emotion-related processing. Additionally, a lot of the early work in investigating the role of the amygdala in fear processing and emotional learning very much informs our understanding of the amygdala today. And today, the research in the affective sciences that's studying the role of the amygdala as it relates to a range of cognitive and affective processes is just continuing to grow. We're seeing this exponential growth in this area of study, and we look forward to learning more about the role of the amygdala and its connection with these other brain regions and a host of other cognitive and affective processes. And with that being said, that's all I have for today.
Thank you very much, Dr. Moreno. That was a lovely talk. It was great to hear about, functioning in terms of how the amygdala functions, getting a good idea of where it's located, which is always important for us to know in terms of our neuroanatomy. And then I really like how you pulled it all together with the case example to really show the impact of when you don't have an amygdala, that fear response is not there. And that's really important for our survival.
Pretty much, yeah.
Very great. Thank you so much. We are going to wrap up now, so please feel free to join us next week. We're going to be talking about the hippocampus, the fornix, mammillary bodies, and memory. That'll be on Monday, July 24th at 2 p.m. Eastern Standard Time. And a recording of Dr. Moreno's lecture will be up later this week, so you can review it as much as you want. Thank you again so much. We really appreciate it.
Great, thank you.